All right, hello everyone. You're tuned into today's PIR live event, and I'm your host for today, Scott Jones. Our guest today is Dr. Thomas Merritt, professor in chemistry and biochemistry at Laurentian University and a CRC in genomics and bioinformatics. Welcome to the PIR live event, Dr. Merritt. Before we get started, I'd also like to welcome today's viewers watching the live stream. It's great to have you with us today, as always. Remember, if at any time you have any questions for Dr. Merritt, you can give us a shout out on Twitter using the hashtag AskPIR. If there's room in your tweet, please include your school name and city, and we can give you a shout out. I look forward to getting to those questions, but first I'll turn it over to Dr. Merritt, who's going to tell us a bit about his background and about his work that he is doing. Very exciting, I'm sure. So go ahead, Dr. Merritt.
Great, thanks for the introduction. Thank you, everybody, for joining us this morning. I'll give you a quick overview of what I do and maybe a little bit about why we do it, and then we'll go to the questions. So I'm a geneticist. At least I was trained as a geneticist, and I really bill myself now as a genomicist. And let's talk a little bit about what the difference between those two things is. So genetics is the study of genes, those packets of information that are inherited generation to generation to generation. And traditionally, we've often thought about genes as these little isolated units. And if you think about the way that we teach genetics in school with things like Mendel's P's and Punnett squares, we often look at genes as these little isolated pieces of information. And that works as a model, but it's very much a model. And genetics is really genomics. Genomes is a genome is all the genes in an organism. So we have about 20,000 genes in the human, so we can think about individual genes, but really what we should be thinking about is how all of those 20,000 genes interact. So what I do as a genomicist is I study how individual genes work, but in the context of all of those 20,000 genes interacting. And that leads to a lot more complicated models and a lot more complicated questions, but it's a little bit more closer to reality than the individual genetics. And we've spent a lot of time as geneticists trying to understand why our models for how things worked, why our understanding just didn't seem to match reality. And one of the reasons it didn't match is we weren't thinking of things in a whole genome basis. And so most geneticists now work in the realm of genomics. We don't work on isolated genes or individual genes. We'll often study one gene, but it's generally in the context of all of these genes interacting in an organism. So I mentioned a little bit about the idea of a genome. That's all the genes in an organism. One of the really interesting things as we've moved into the realm of genetics or into the world of realm of genomics or the world of genomics is understanding better just how many genes there are and how those genes interact. So prior to sequencing the human genome, so we've now read the information in the human genome, and we've read that for now really hundreds, probably thousands of individuals. Before we had that genome sequence, we had estimated that there were probably about between 80 and 100,000 genes in the human genome to give us the complexity that we are. So we understand quite a bit about our biology. We thought we understood quite a bit about how genes worked. And our guess as to how you would connect from information to biology was that was going to take about 80,000 genes to do it. And one of the first really surprising results from genomics when we sequenced the human genome is that there are fewer than 22,000 genes in the human genome. And that may not seem like a big deal, but that's a pretty big miss. If you think that there are 100,000 genes or even 80,000 genes in an organism, and us, I mean, this is not an esoteric organism, this is us, humans that we've been studying for an extremely long period of time, and you're guessing that we've got about 80,000 genes, and you find out that there are 20,000 genes, something is drastically wrong with the way that we're thinking about genes. And what we've spent the last 10 years doing is trying to understand what was wrong in our first estimates of how genes worked that led us to that mistake, right? That misestimate of 80,000 instead of 20,000. And the first piece that came out of it was that what we were missing really fundamentally was just how complicated individual genes are. So genes are these packets of information in almost all organisms on the planet. Genes are DNA. What we didn't understand was just how complicated those individual genes are. And what we've done for the last 10 years is try to understand how those genes interact in a more complicated fashion to give us the complicated biology that we see. Now, I'm a genomicist. I work on genes. I work on DNA. I specifically work on fruit flies. And this is a vial of fruit flies. These are some flies I actually stole from one of my graduate students. I have to give them back before they realize that they're gone. But this is the same fruit fly that flies around your fruit bowl. That is literally the same fruit fly that flies around your fruit bowl. And some of the flies that we work on in the lab are from the compost pile in my backyard. So why work on flies? If what we're interested in is genetic complexity and we want to understand the human genome and the complexity of human genetics, why would we work on flies? Well, part of the answer to that is the idea that life on this planet probably evolved once in a fairly complicated pattern, but life started as relatively simple, single-celled organisms and then expanded out from that with a lot of crosstalk and back and forth. But the genetics of this fly, or these flies, is identical to the genetics in humans. And the way that the fly genes work is exactly the same way that human genes work. And that's because we share this common evolutionary history. So there are a lot of questions that we'd like to answer in humans, but we never ask the question in humans. Ethically, we wouldn't conduct that. So genetic engineering is a really powerful tool to go in and modify individual genes, change the way a gene works, to try to understand what the role of that gene in the organism is. We can do that in flies in a way that we would never do in humans. They're easy to grow in the lab. We can get samples pretty much anywhere that humans are found. We can find flies. The biochemistry is very similar. The genetics is very similar. And they're a really great tool for us to work on. They're also really tiny. So this is a rack of flies. This is a rack of flies that are actually on my desk. This rack will hold about 8,000 flies. One of the really interesting questions in genetics is individual variation. So if you're in a classroom now and you look around that classroom, we're all very distinct. We're different heights, different weights. Our behavior is different. The way that we learn things, the way that we interact with our peers, those are all really different characteristics. And all of those have a genetic component. There's a genetic piece of each of those things. If we want to understand genetics, one of the questions that we have to really address is this genetics of individuality. And one of the ways that we can use the fly model is to address that. When we're looking at a rack as small as this, that can hold 8,000 individuals, we can go in and ask a lot of really interesting questions about how those individuals vary. And because they're flies, we can go in and genetically engineer them. We know there's the entire genome. There are a lot of tools that we can use to get at those questions. So the flies are a really great model, and because of this shared evolutionary history, they allow us to expand what we know about humans by addressing those questions in the fly itself. So we mentioned ways that we use the fly. A lot of the work that we do in my lab will go in and we'll mutate a gene, or we'll find a natural variant of the gene, and we look to see what happens to that fly, what happens to the biology, how do you connect between a gene and some biological characteristic. One of the other things that we can do is use flies to understand metabolism and physiology of humans. And so we've started doing some work where we're looking at the physiology of deep mining. So working deep underground, there are a lot of challenges, physical challenges to working deep underground. One simple way to think about that is as you go deeper underground, the atmospheric pressure gets higher and higher just because of that column of atmosphere on top of you. So in the last year or so, we've started a project using fruit fly and using a deep underground mine to understand the physiology and metabolism of working deep underground. And we can do that because of all the tools that we have for flies. And we do that up here in Sudbury using the Snow Lab at the bottom of Crichton Mine. And the Snow Lab is famous now for hosting or housing the Nobel Prize winning research on neutrinos. It happens to also be 2 kilometers underground. And that position 2 kilometers underground, Scott, as you know, makes it uniquely situated to study neutrinos and the way that these fundamental particles make up the world around us. It also is an incredible lab for us to do fly genomics and study fly metabolism, looking at pressure deep underground. And so we've been taking flies down to Snow Lab and doing what we call metabolomics to understand the fundamentals of metabolism using the fly model and using this incredible deep mine lab in a way that really was not necessarily initially planned. So one of the neat things about science is you find an interesting question and find interesting ways to address it. And that can lead to things like using the fruit fly that flies around your fruit bowl in the summer to understand the physiology of working deep underground by taking advantage of a world-class physics lab. So that's sort of the background that I wanted to give on the kind of stuff that we do, the genomics, genetics. I talked a little bit about metabolomics, which is looking at metabolites, thousands of individual small molecules, sugars, proteins, amino acids that make up an organism. Maybe we can go and see if there are any questions about the kind of work that we're talking about.
Sure. So let me just take a quick look here. So one question I was interested to ask is why we found a quarter as many genes as expected in the human genome, and how flies help us to understand that. So it's kind of not obvious to us that flies would be so genetically related to humans, right? And you said it's not simply the case that one gene affects one phenotype in one particular way, right? There's some interconnected things there going on.
Yeah. So those are really two awesome questions. So the first question is, why were we so far off on estimating the number of genes? And then the second question is, what can flies tell us about that? So let me hit the first question first. What we had missed was just how complicated individual genes are. And so we had a really simple model where you have essentially one gene, one protein. And to get to the complexity of us, that the complexity of our biology, our metabolism, our physiology required that there would be about 100,000 genes to do that. If you take this simple model of one gene, one protein, it ends up that most genes, it's not that simple. You've got one gene, you've got multiple proteins, or you've got one gene and one protein, but the protein is only on in some tissues. And basically what it comes down to is we didn't understand how complicated genes were regulated. So I'll give you an example. There are about 20,000 genes in the human genome. In any given cell, only about 2,000 of those genes are working. All right, so you've got this book, and the genome, all of those genes, is essentially this cookbook, this encyclopedia, this dictionary. It's a reference. Every single gene is present in every single tissue. What makes one tissue distinct from another, so what makes a nerve cell, a muscle cell, a muscle cell, isn't what genes are present, but what genes are active, what genes are turned on. And so what we had missed in this misestimate of the number of genes was just how complicated that regulation was. So we now understand that it's not a lot of genes, it's fewer genes with a really complicated pattern of regulation, turning these genes on or turning these genes off. So how do we use flies? It ends up that in flies there are about 15,000 genes. And one of the things that we do in my lab is we can use the tools that we have in flies to understand how genes are regulated. What turns a gene on, what turns a gene off, what turns a gene on in one tissue but not another tissue. So that study of regulation, to do that, you have to be able to take the system apart, break pieces out, put pieces back in, experimentally manipulate the system that you're working on. And to do that kind of experimental manipulation, you want something other than humans. You don't want to be taking apart the human genome to understand how it works. So you take apart the fly genome and you piece it back together, and you do things in flies that you would never ethically do in humans to understand how humans work. And it's the same because of that shared evolutionary history. The way that genes work in flies is very similar to, and in many cases, exactly the same as the way that genes work in humans.
Okay, so a bit on the flies then. So I get the whole idea is you're affecting the genome of the fly to see what effect that has on their biological characteristics. But how does that actually work? Like, how do you get the DNA out of the fly, and how do you keep track of which fly you've affected and which you haven't, and some of the details that way?
Those are great questions, and it can differ depending on the question that you're asking. So some of the times what we really want to know is an individual fly. And so we'll take a vial like this, and we know the genotype of these flies. So we have sequenced the DNA, we've read the DNA from the flies, we know particular genes that are present or absent. And in some experiments, what we do is we take all of these flies that are multiple different genotypes. We can take a single fly, one fly, which a fly weighs about 1,000th of a gram. We can take that one fly and 200 of its brothers and sisters out of a vial, and we can study the biochemistry of each of those flies individually. So you take a fly out, you grind the fly up, and we can do enzyme kinetics, simple biochemistry, on those individual flies and then tie that back to the genotype. Sometimes all of the flies in a vial are very similar, and so what we'll have is not that we're looking at the 200 flies that are here, but we'll have groups of, say, 10 flies or 20 flies, and we've got 20 vials. And what we want to know is how is one vial different from another vial, different from another vial. So depending on the kind of questions that you're going to ask, you can be looking at individual flies, you can be looking at sets of flies. Getting to the biological material, getting to the enzymes, getting to the DNA is really as simple as grinding them up. So I don't know if you've ever done the experiment, but you can take bananas and mash bananas up in detergent and salt and actually extract out the DNA. You can do the same thing with human cheek cells. Extracting DNA ends up being really easy. Sequencing the DNA is getting easier and easier. The really complicated thing is how do you go from that DNA sequence to the complex biology that's out there. So you and I have about 9 billion pieces of DNA, units of DNA, 9 billion letters in our genome. There are about 6 million differences. A very small subset of those differences are the reason that you have dark hair and I have slightly lighter hair. Or what color are your eyes?
Brown.
Brown. Okay, so eye color is awesome. So we often think about eye color in a really simple way, and you've got brown eyes and blue eyes, and there's a gene that determines brown and blue. There are at least 14 other genes that interact with that gene to give you different colors. So if you look at my daughter's eyes, she has blue eyes, but they're a slightly different blue than mine, and my wife's eyes are green. And the genes that are involved to give us those subtle differences in eye color interact to give us different colors. That is a really great model for how the genetics of disease work. So 1 in 5 Canadians are going to get cancer. Every one of those Canadians is likely to undergo chemotherapy, and each individual will respond to that chemotherapy differently. What we would like to do is understand genetically what drives some of that difference. Can you predict whether somebody is going to respond to this type of chemotherapy or that type of chemotherapy? We're not there yet, but we're beginning to understand the genetics behind that. And one of the ways that we can do that is study the genetics of individuality using things like flies.
Right. Well, that's very interesting. We've just had a great question come in over Twitter here again from, looks like Glen Forest Secondary School in Mississauga. And they're asking, what are the effects on the metabolism of the fruit flies while they were underground in the snow lab?
Yeah, that's a really neat question. So what we're doing with the flies in Snow Lab is we're taking 3 different types of flies. They're all the same species, but they're different lines. And essentially, they're 3 different families of flies. And we're taking them down to Snow Lab, and then we're bringing them back up. And what we want to know is how each of those families respond and how they respond differently and how they respond similarly. So what are the common things that are across those 3 families and what are different? So we look at about 2,000 individual metabolites. And metabolites are the things like sugars, lipids, amino acids, the small molecules that make us, us. And when we quantify those 2,000 metabolites, we found that about 10% of those metabolites differ from a single trip underground. So you go 2 kilometers underground, it's about a 20% increase in atmospheric pressure, and about 10% of the metabolome changes. Now, the tricky thing with metabolomics is we can identify the presence of 2,000 metabolites, but we can only name a few hundred. And so what we're doing now is trying to figure out not just that 10% of the metabolite change or metabolome changes, but what 10%? And what we'd like to be able to do is start to look at pathways. So is there a particular pathway? Is protein synthesis turned up by high pressure? Is it turned down by high pressure? And our ultimate goal is to try to understand what is going on with what's going on in metabolism, what's the physiological stress of working deep underground, and then can we work with that physiology to make working underground, working at high pressure, safer, easier, less stressful on the organism. So the project is very much in its infancy. The short answer is about 10% of the metabolites that we can measure change, but we're not yet to the point where we can say this pathway is upregulated or this pathway is downregulated. But to put that in context, if you expose flies to pesticides, about 30% of the metabolome changes. So what we can really define as a highly stressful environment, exposing a fly to a pesticide, changes about 1 in 3 metabolites. Simply going down to a deep mine and coming back up changes 1 in 10. So that seems like a really substantial change. The question we're going to get to now is there a way to make those changes or to make that stress less stressful? Is there a way to make mining healthier by understanding how physiology is changing?
Okay. And say that some metabolic or some genomic change were to occur in some extreme environment like that, would these effects be long-lasting, or would they just be temporary effects? What's the time scale on those?
Yeah, that's a great question. So what's the time scale? Are these short-term or are they long-term? We don't know. We literally have just started scratching at this. We've got an experiment going down to Snow Lab on Monday, where instead of just taking flies down once and bringing them back up, we're going to take flies up and down for a series of trips over the course of a week, and we're going to look to see what happens there. There's some other really simple experiments that come to mind where you could take flies, leave them at pressure. What we're trying to see what happens over a period of time, right? So if you put a fly at high pressure for a week, does that spike and change in metabolism start to drop off? We don't know. We can ask that. What we're trying to do with this particular project is apply it to the way that deep mining works. And in deep mining, a miner goes down to depth, comes back up, goes down to depth, comes back up, and does it over their working lifetime. So it's not a matter of going to a deep mine and then staying there. It's the up and down. And we are suspicious that the physiology, the metabolism of that stress has to do with going from normal pressure to high pressure, to normal pressure to high pressure. And so that's where we're focusing our questions now. When you take a fly and go up and down, what happens to that fly? One of the neat things about flies is they're relatively short-lived. So a fly only lives about 30 days. So if we take a fly and we bring it down to Snow Lab over the course of a week, that's really similar to what a miner would experience working deep underground in their working lifetime.
Cool. So yeah, very useful to people who would be in the industry for sure. I guess it's kind of you talk a bit about genetic mutations, and that's kind of like we think we get superpowers after we undergo genetic mutations or something. But being that that's essentially what you're doing, how often would that occur if you weren't involved? Like, how often does a genetic mutation just occur randomly? Yeah.
So what's the frequency of genetic mutations? That's a really good question. And there's a lot of work by a number of different labs trying to look at that. One way to put that in perspective is to think about how genetically different all of us are. And so, yeah, we do think of, you're going to get superpowers, and you're going to get telekinesis, and you're going to be Cyclops or Professor X, or you're going to sprout wings. I can tell you really straightforward, you are unlikely to sprout wings. As much as it would be super cool to get wings, you're not going to grow wings. What we do know about mutation is that the way that we think about sort of either normal or mutant is wrong. So any two individuals, so if you're sitting in a classroom and you've got 20 people in that classroom, you are all 20 of you genetically distinct. There is no one human genome sequence. Every individual has a unique sequence. And so mutations are really the norm. So each of those differences is a mutation, but everybody is a series of mutants. So we're all mutants. There is no wild type. There is no normal. The normal is that we're all very genetically distinct. One of the really neat things in genetics is we're beginning to understand not just how common those mutations are, but different types of mutations. So in my daughter's SK class right now, they're a pair of identical twins. And you might think of identical twins as being, the example of individuals that are genetically identical, and they're not. They're actually genetically distinct. And what's really cool is they become more genetically distinct over time. And the DNA sequence doesn't change, but chemical modifications, what we call epigenetics, beyond standard genetics, epigenetics, that epigenetic change changes. And so two identical twins can have different epigenetic markers, these chemical modifications to DNA, and those differences get bigger and bigger through time. So even two genetically identical individuals aren't identical, and as they get older, they get more and more distinct, more and more different. One of the really fundamental changes in biology since or changes in genomics, along with understanding just how few genes we have and how complicated those genes are, has been understanding how important this epigenetic modification of that DNA really is.
Right. Okay, take another look over at Twitter here. We've got a question from Rachel Tran. I'm not sure which school she's at, but she wants to know how the flies that you use and that you're doing the tests with, how do you know that they were all in the same condition to begin with? Do you breed them right in the lab then, or?
Yeah, that's a great question. So what's our baseline? What's our control? How do we do that? We take a lot of steps to try to make the flies the same. So we actually go through things, and one of the reasons that we can use flies is we can inbreed flies. So within a vial of flies, all of these individuals can be very genetically identical. Now that sort of goes against what I just told you, with everything being slightly genetically unique. But within those limitations, we can create flies where this entire vial is genetically identical. And we can set things up where we know the parents, we have controlled mating of the parents, we create a common density of flies when they're growing, we grow them in a controlled environment, so that within that vial, flies are as identical as possible. The next thing that we do is we very rarely work on individuals. So I mentioned the idea that we can do experiments where we're pulling out individuals, but what we generally tend to do is we'll take a couple of hundred flies in a vial like this, we'll put them in groups of 10 or 20 flies, and each of those flies is slightly different, but then we try to get an average effect across that, and then what we end up looking at is dozens of different vials, or half a dozen different genotypes, different families of flies, to try to average out those individual differences that we see. So there are a lot of things you can do in the lab. You grow them in a set temperature. It's usually 25 degrees. You grow them in a very moderate humidity that you can regulate in the lab. You grow them in a controlled light cycle. So if you change the light-dark pattern that the flies are in, you change the metabolism of the fly. So all of our flies are grown at 25 degrees Celsius in an incubator that has lights that are on for 12 hours and off for 12 hours. If you change any one of those pieces, even if you change the humidity, but certainly if you change the light cycle, if you change the temperature of the fly, the physiology, the biology, the metabolism of that fly changes, and often changes in interesting ways. So we can ask questions about how that physiology changes.
Yeah, like you've already told us, the environment is very important, right? So we want to make sure that they're all starting out in the same environment, right?
Absolutely.
So I see, as I introduced you, that you're a Canada Research Chair in Genomics and Bioinformatics. So the bioinformatics part, is it essentially the goal to basically take everything from the lab and make it a math problem? Like once you know the connections between this gene and that gene and see how they're all related that way? Is that the goal with the bioinformatics part?
That might be one way to look at it. So bioinformatics is simply processing of large amounts of biological data, often large data sets, using computer science. And if you think about the way that we are thinking about genetics, we're no longer thinking about this gene, this gene, this gene. We're thinking about these 20,000 genes and how they interact. And so a lot of bioinformatics is the computer analysis of these enormous data sets. So when I was a graduate student, I was working on a small family of genes, and I was working with maybe 15 or 20 gene sequences to do an analysis. It's not quite a fair comparison, but one of the studies that we're doing in my lab right now, looking at diet of a particular fish-eating bird, and we've got something like 5 million individual DNA sequences that we're working on. So a lot of bioinformatics is just, to some extent, mathematically, but computationally, how do you gear up from a case where you're doing a comparison of 15 genes to a case where you're now looking at 5 million genes that you've generated in less than a week in the lab? So the amount of biological data that we create is just astronomical, and that is driving computer science to come up with better algorithms to process that data.
Okay, so we don't have too much time left. I wanted to squeeze in the question that I ask all of our guests, and that is getting back to a bit of your background. What initially got you interested in the studies of genomics and genetics, and what advice would you have for K-12 students who are looking to maybe get into that field in the future?
Sure. I think, like most biologists, I was, as a kid, you're just sort of fascinated by the world around you. So I spent a lot of time out in the woods chasing turtles, chasing snakes, and that sort of fascination with the biological world led me to try to ask some questions about what would drive that. So if a particular turtle species is only found in one environment or another, what is it that makes that turtle well suited to one environment? Why are two turtles different? And turtles are sort of tough to work on. They live a long time. They don't particularly like to be bred. They don't work well in vials. So when I started asking questions about biological complexity, biological diversity, I started looking for systems that I could work in. And it ends up the fruit fly, because they're tiny, they're easy to breed, they're easy to relatively easy to genetically engineer, allowed me to ask some questions. So what I would suggest to budding young scientists is be curious. One of the best pieces of advice that I had from my advisor when I was a postdoc was to look for things that don't make sense. And so you get a set of experiments and you would like them to be neat and tidy because then you can write everything up and it'll be fine. But the really fun things that come out of science are the things that don't make sense. And what really defines modern genetics, modern genomics, is that we're finding so many things that we had wrong in our simple models, and we're trying to figure out more complicated models that actually allow us to address those. So follow your curiosity, look for things that stand out, look for things, little glitches in the matrix that don't make sense, and follow those, because those are often the most exciting things that you get to study. Genetics, genomics is amazing. We get to understand the biology, the fundamental biological process that drives all of us. And every day in the lab, I get to talk to my students about, what have we found today? It's a neat job to have. It's an exciting place to be. There are lots of changes going on in genomics, genetics, bioinformatics right now.
Right. And I think it's the case that we're often wrong in science, right? But it's just a matter of figuring out why we're wrong and then how to figure out what that tells us being wrong, right?
That's absolutely. And I think one of the best things that you can do as a student is understand that most things that are in your textbook are not correct, or at least they're not complete. And the exciting thing is getting to be part of understanding what did we have wrong on this model, what was incomplete, where did we miss this or that. How did we get so wrong with estimating there were 80,000 genes when there were only 20,000? Answering that question is leading to some really neat things, better understanding of the way that genes actually work. And it's a lot of fun to be part of that.
Very good. Well, looking at the time, I think that's actually, unfortunately, all the time we have for today. So I'd like to thank you, Dr. Merritt, for taking the time to answer our questions, and thank you for all of the viewers for tuning in and sending the questions. I'll give a quick shout out to our next live event, which is next week, January 27th at 11 a.m. with Dr. Ning Yan from the University of Toronto. Her research is on forestry-based alternatives to petroleum-based materials and the biorefinery project. So we'll ask her lots of questions about that at that time. So thank you, Dr. Merritt, again.
Thanks, Scott. I appreciate you having me, and thanks for everybody for tuning in. It's been a lot of fun.
Very good. We'll see you all next week.